Okay, so now we're moving on to our next activity. We're going to talk about DNA as a library. So, what is a library? Or what's in a library per se?
Books.
Books. And what do you use books for?
Research.
Research.
Reading.
Reading. Information. Okay, good. So now, with that being said, I want you to think about the nucleus is like a library. So, what is the nucleus?
The library.
No, like what is the actual nucleus? What is it in biology? It contains the center of the cell. It contains what? The DNA. The DNA. Okay, and what does that help us do?
Grow.
Grow.
Function basic activity.
Function with the basic activities. Good. All right. So in front of you, you have a few questions you're going to answer. Based off of the nucleus as a library. And I want you to think about how DNA functions in the cell and answer the following questions below. So, first question, what is DNA similar to in the library? So we have different components of the library. What would the DNA be similar to in a library? You guys already told me books and other things. Number two, why is there control over some of the information in the library? So it's not like you can just go and grab everything and be like, all right, this is what I'm taking, but there's some stuff where like you actually have to check out. It's not just easily accessible to you. And then three, how can the information that is not allowed to leave the library still leave? Those are your three questions. And I'm gonna give you guys a few minutes to answer those. Okay, so if you're not done, it's okay. We'll give you a few minutes to turn and talk, well, not minutes, but a few seconds to turn and talk with your group, and then we'll come together as a whole group and discuss. So go ahead, you can now turn and talk. Okay, bring it back in 3, 2, 1. Alright, so I walked around, I heard everybody share, and I actually read some before I had you share. And if I put a black dot next to your either 1, 2, or 3, that means I want you to share that. So, number 1, what is DNA similar to in a library? Nigeria?
I said DNA is similar to a library because they both hold important information.
Very nice. Number 2, why is there control over some of the information in the library? Sanaya, was that yours?
I said to ensure proper regulations and prevent misuse.
Proper regulations and issues, really good. And then number 3, how can the information that is not allowed to leave the library still leave, Kendrick?
If something were to escape from the library, for example, information could be lost. Another way is if specific permissions were granted to let that information leave.
Yeah, so we can have some information lost. Maybe there's some information that needs to go one place and not the other. So kind of the same with libraries. So I know here where we're from, it's not a lot of stuff that you can just check out. But like in bigger libraries, there are some books that like you can only see them in the library. Like you have to put in a little request in paper and bring the book to you. Then you have to turn it back in before you leave. So that's like a big library in London. Alright, we're gonna do our next, well no, let me do our next part. So here is kind of an example more in depth of DNA like a library. So, we have the letters. So the letters could be our nitrogen bases. And what are our nitrogen bases? We learned them yesterday.
ATGC.
ATGC. Alright, then we have the words. That's the genetic codes created using the nitrogen bases. So this is like basically inside from the library going out. Then we have our books. That's our genes that are made up of a variety of genetic codes. And then we have the library as a whole, which are the chromosomes that contains the genes. So this is like kind of, we broke the library down piece by piece. Alright. So next, I'm gonna give you guys an article. Don't worry, you're not gonna read the whole thing. We're gonna do jigsaw. So whatever part that's in purple, that's the part that you read, and then we're gonna come together and we're gonna share. So you can write on these, you can underline. If you have highlighters, you can use highlighters and highlight. And then we'll share each important part from our articles. You only need the purple part. So like you only gotta read these 2 paragraphs, right? And then we're gonna come together. No, you don't have to read out loud, you read it individually. And then you annotate your notes and then you'll come out loud and we'll share as a whole group what you learned from your part of the section. So yes, these are yours, you can write on them. Dr. Hilbert's office. Who's that?
Graduation.
And when you're done. Alright, a few more minutes, and you'll get ready to share your part of the article. Okay, we ready? Alright, so now I just want you to share your part of your article with the class. And I'm doing it in the order that I had it highlighted, so first, second, third, fourth, fifth. Alright, so first, D'Angelo, you had the first two paragraphs. What did you gather from your part of the article?
One thing that I did get was in 1869, it was landmark of the year, Geneva research.
So that's when they started the
Yeah, this one
research?
started research.
Alright, and it's 69. Alright, Kyria, I think you have the second part of the article.
Something that I took away from my section of the article was that Phobos Levine discovered the three major components of a single nucleotide.
right? Did it tell you what they are?
Yes. Phosphate, sugar, and the base.
Alright, you got the phosphate, the sugar, and the base. Alright, so now you were next.
Okay, one important thing that I got was each nucleotide has 3 components: a phosphate group, which either includes ribose or
Deoxyribonucleic.
and it has a sugar and a single nitrogen base, and that the RNA contains A, G, C, and A, G, C, U, and the DNA contains A, C, G, T.
So one has U and one has what?
T.
T. Alright, good. Jessica had to step out, but her section was just more so about how like the math adds up. So even though A and T pair, when you see A and G, they'll have about the same amount of periods. And then same for C and T, they'll have the same amount of periods to equal out the math part, but we don't have to go that in depth, so it's okay. And then Kendrick, you have the last part of the article.
Short there was that A and T connected and C and G helped them figure out the shape of DNA. So how it's double helix, 2 of them were right-handed, while the other one, Z DNA, rotates leftwards.
Alright, and that was just more so the structure and the function, of the DNA. Alright, good. Alright, so our next part, using your evidence from your article and what you heard your classmates say, we're going to do a gallery walk. So I'm going to give each of you guys about a minute at each station, and you have your marker, and you're just going to answer the question. You can also put any comments you may have about the question, or like if you're not sure what questions you would want to ask me. So basically, you can be either one or all 3. Does that make sense? Alright. So, go ahead and start. Everybody should be at each station. There's 4 different questions, and I'm going to read those out to you. So, over here, how does the structure of DNA ensure accurate replication? Right here, we have when does DNA replicate? What will happen if our DNA doesn't replicate correctly? And then what do you think was the most significant challenge in discovering the DNA structures? Those are your 4 questions you're going to answer. And you'll have a minute at each station. Alright, D'Angelo, once you get there, I'll start the minute. And your time starts now. So again, you can write what you think, you can add a comment, answer the question. If you have a question based on the question, do y'all need more time?
Yeah.
Okay, I'm gonna add another minute. Alright, and remember, you're using your evidence from your article. one more minute? Okay. You just have a question or a comment,
Of course, yeah, I got that.
but you only have 34 seconds, so you don't have 12 seconds.
We all come to this section. Like, we all got it.
Finish your Alright. Okay, rotate this way, this way, this way. Alright, I'm gonna put 2 minutes on the clock this time. You got that one? Okay, ready? Go. Try not to write what the person before you wrote either. And again, remember, you're using evidence from your article. That's why you have your articles while you're rotating. Use your evidence to answer the question. You can also add on to the person before you. Just don't write the same thing. Like if you agree with what they're saying and you want to add something onto their thought, that's fine as well. I'm gonna give y'all a few more seconds, so I still see 3 people writing. Alright, rotate this way, similar done, rotate. Alright, ready, go. Alright, ready, and y'all can start. Alright, make sure you're reading what your classmates said before you. Don't write the same thing. You can add on to it though, just don't put the exact same thing. And then also remember, you can use the evidence from your article. Use the evidence from your article. You got 1 more minute. 1 more minute. Make sure you're using the evidence from your articles to help with your responses. Okay, that is time. Alright, so, nope, stay where you at. Stay where you at. Alright, we're gonna go around and we're gonna rotate again, but this time we're gonna talk about the responses on each paper. But when you're at the wall, I want you to read someone else's response and I want you to tell me your thoughts on their response. Okay? So, let me start with you, Kendrick. Which one do you agree or disagree with? And then tell why. Or if you want to answer one of the questions, because there's a lot of questions over here for the where it says what will happen if our DNA doesn't replicate correctly.
For the top question, it says, are there any syndromes or sicknesses from it? There are several syndromes or sicknesses that happens when DNA does not replicate correctly, like Down syndrome, Edwards syndrome, which is where somehow the DNA malfunctions and chromosomes, which are part of the DNA, get set to a little too extra to one chromosome or a little too little, or not enough to another chromosome. So yeah, there are several syndromes where the DNA doesn't replicate.
Very good, very good. D'Angelo, which one do you agree with or disagree with from your side?
I agree with. I think the most significant challenge in discovering DNA structure was trying to figure out which chemical base went with which, or coming up with specific names for everything.
Okay. Criteria. Which one do you agree or disagree with? Not yours though.
I agree with the structure of DNA makes unraveling and reading the genome sample. In the article, 3 formations were discovered for different purposes. The 3 forms are aDNA, bDNA, and zDNA.
Okay, and you read that in the article as well? That's a really good one. Good job using that evidence from the article, Samaya. I agree with all of them. Why?
Because they all basically was talking about the same thing, mitosis and meiosis, DNA replicates when you're trying to make a person or
Alright. And I remember, that one was D'Angelo's, the new body part. So his example was like, if you fall, and like, let's say you scrape your skin, you know that is part of your DNA, but it grows back, it grows over. That's a type of DNA replication known as mitosis or meiosis. Mitosis. Remember, meiosis makes me, so meiosis is like babies. Mitosis makes mitosis, so like your new skin cells and all that stuff, that's mitosis. All right. Good job. Good job. You can head back to y'all seats. I just want to touch on what will happen if DNA doesn't replicate correctly. I also want you guys to know that like you get mutations, but not all mutations are bad. So the one I like to tell everybody is dimples. Dimples is a mutation, like that's DNA. Something happened and that's okay. That's a not good mutation, but it's not bad. Alright, any questions? Alright, go ahead and get your other sheet of paper. We're going to start on our closure for today.